PowerPoint folder which I have shared with you as well as with regular CBSE group. Okay. So in today's topic, we'll be discussing about the biomicromolecules, sugars or carbohydrates in general. We'll be discussing about that. So you know that sugars are sweet and they are water soluble carbohydrates. So, and unlike the lipids which are water insoluble, sugars are sweet and they are water soluble carbohydrates. So they are formed of carbon, hydrogen, and oxygen in the ratio of 1 is to 2 is to 1. Example, glucose C6H12O6. So you can notice that ratio of carbon is 1 and oxygen is 1, whereas hydrogen is 2. 1 is to 2 is to 1 ratio you can notice in case of glucose structure C6H12O6. So you notice this glucose structure will be having that. So it is a hexane ring, and you notice they would be having the OH group and CH2OH group apart from this carbonyl group. Okay, so the glucose C6H12O6. Then ribose sugar is C5H10O5. So the again you notice here 1 is to 2 is to 1. So ribose is a pentose sugar, and you can notice that they have this formula of C5H10O5. So these are the types of sugars which have the carbon, hydrogen, and oxygen in the ratio of 1 is to 2 is to 1. So the glucose and the ribose sugar. So this is what is the formula of them. Okay, and the structural formula of glucose, which has CH2OH as well as many OH groups with oxygen, the carbonyl group. Even here, you notice that CH2OH as well as the many OH groups with carbonyl groups in ribose. So this is the structure of a sugar. Examples are glucose and ribose. So the next biomicromolecules that we'll be discussing about is the nitrogen bases. So nitrogen bases are found in the nucleic acid like RNA and DNA. So nitrogen bases, these are the nitrogen containing cyclic compounds found in nucleic acids. So nitrogen bases are nitrogen containing cyclic compounds found in nucleic acids. So they are classified as purines and pyrimidines. So purines we can remember AG, and pyrimidines you can remember it as cut. So purines, AG, adenine and guanine, and pyrimidines, you can remember it as cut, CUT. See, the pyrimidines, they have a single ring structure, whereas purines, they have a, uh, they have a exos with pentose ring structure. So whereas pyrimidines, they have only the exo structure. Okay, so the nitrogen bases are of two types, purines and pyrimidines. Purines consists of two type of nitrogen bases, that is adenine and guanine, AG we can remember, and pyrimidines consists of CUT, that is cytosine, thymine and uracil. CUT you can remember cytosine, uracil and thymine. Uracil is found only in ribose nucleic acid, whereas thymine is found only in the deoxyribonucleic acid. So nitrogen bases are the nitrogen containing cycle compounds found in nucleic acid. They are classified as follows. Nitrogen bases are classified as purines and pyrimidines. So purines consists of adenine, guanine, and pyrimidines consists of cytosine, thymine, and uracil. So pyrimidines consists of cytosine, thymine, and uracil. So purines have a two rings they are having. One is the exos and the other is the pentose part of it, whereas the pyrimidines they have only the uh, exos ring structure. So that is a few of the points that you can remember about. So we have the example of adenine, which has the amino group and nitrogens. You can see the nitrogen containing bases, we call it as containing cyclic compounds found in nucleic acids. So the nitrogen base adenine, which is a purine, having a exose and pentose sugar together. So two rings they are having it, whereas the pyrimidines, example uracil, you notice that they have only a single ring. Okay. The next slide is about the nucleosides. See, nitrogenous bases plus sugar, we call it as nucleosides. Nucleotides are nitrogenous base plus sugar plus phosphate group. We call it as nucleotide or base pairs, what we call it as nucleotides. Okay, so these are the nucleosides, are nitrogenous base plus sugar. So these are the compounds formed by the linking of a nitrogen base with a sugar. Like you have nitrogenous base plus sugar is nucleoside, adenine plus sugar is adenosine. If it is ribose, we call it as ribose adenosine, R adenosine. Guanine plus sugar is guanosine. Cytosine plus sugar is cytidine. Thymine plus sugar is thymidine. Similarly, uracil plus sugar is uridine. So the nucleosides are nitrogenous base plus sugar. We call it as nucleoside. So nucleoside is composed of nitrogenous base and sugar. So these are the compounds. Nucleosides are the compounds formed by the linking of nitrogen base with a sugar. So adenosine. So they have an adenine group attached to this ribose sugar or deoxyribose sugar. So uridine, you find it in case of uridine is noticed in case of ribose sugar. So you can see the uracil is attached to the sugar. Okay, and you find only in ribose nucleic acid. Adenine you find both in ribose nucleic acid as well as the deoxyribose nucleic acid. So they have this uridine is uh, uracil plus sugar. So they are found attached to the sugar. So nitrogenous base plus sugar, we call it as nucleoside. Similarly, adenine plus sugar, adenosine. So the guanine plus sugar, we call it as guanosine. Cytosine plus sugar is cytidine. The thymine plus sugar is thymidine. Uracil plus sugar, we call it as uridine. These are all the examples for the nucleosides, the types of nucleosides. And here you can see the structural formula of a sugar having adenine. So adenine plus sugar is adenosine. So this is adenosine. Seeing it itself, you should be able to say that. Uracil with the sugar. So we can immediately call it as uridine. So uracil plus sugar, we call it as uridine. So these are the structures of nucleosides that you have to remember about. Then the fourth one is nucleotides. So what are nucleotides? These are the compounds formed by linking of a nucleoside, that is nitrogenous base plus sugar with a phosphate. So the nitrogenous base plus sugar plus phosphate, we call it as nucleotide, or you can also call it as nucleoside plus phosphate is nucleotide. Okay, so nucleotides are the compounds formed by linking of a nucleoside, that is nitrogenous base and sugar with a phosphate group. So we call it as nucleotide. So now you can see in this example, so the sugar which is having this adenine, so adenosine, but since they have a phosphate group also attached here at the CH2OH, so you find the H is replaced by the phosphate group. So that is H2PO3. So the phosphate group is attached to the CH2O part of it, and H is replaced by the phosphate group. So we call this nucleoside as adenylate acid. So nucleotide is nitrogenous base plus sugar plus phosphate group. So it can also be defined as nucleoside plus phosphate group. So the example is adenine plus sugar plus phosphate group. We call it as adenylic acid, or you can also call it as monoadenosine, adenosine monophosphate, AMP. We can also call it as. Okay. The next one is guanine plus sugar plus phosphate, guanilic acid, or we can also call it as guanosine monophosphate. Okay. Cytosine plus sugar plus phosphate, we can also call it as cytidilic acid or cytosine monophosphate, CMP. So adenilic acid or AMP, 5 AMP, guanilic acid or 5 GMP, cytidilic acid or 5 CMP, thymidilic acid or 5 TMP, uridylic acid, or we can also call it as uh, UMP. So adenilic acid or AMP, guanilic acid or GMP, cytidilic acid or CMP, thymidilic acid or TMP, uridylic acid or UMP. So where UMP means uridine monophosphate, AMP means adenosine monophosphate. So you can also call, instead of calling it as adenilic acid, you can also call it as adenosine monophosphate. So these terms are not given here, but you should be aware about that. So nucleotides are compounds which are formed by linking of a nucleoside, that is adenine plus sugar plus phosphate group. So adenine and sugar plus phosphate group with a phosphate nucleoside with a phosphate or nitrogenous base. It is composed of nitrogenous base, sugar, and phosphate group. So that is what is the definition of nucleotide. So adenine plus sugar plus phosphate, we call it as adenilic acid or AMP. Guanine plus sugar plus phosphate, we call it as guanilic acid or GMP. Cytosine plus sugar plus phosphate, we can call it as cytidilic acid or CMP. Thymine plus sugar plus phosphate, we can call it as thymidilic acid or TMP. Uracil plus sugar plus phosphate, we call it as uridylic acid or UMP. See, UMP is noticed only in case of ribose nucleic acid. Thymidilic acid is noticed only in case of deoxyribonucleic acid or DNA. So the remaining adenilic acid, guanilic acid, or cytidilic acid, they are noticed in both DNA and RNA. But uridylic acid or UMP is noticed only in RNA. Thymidilic acid or TMP is noticed only in case of DNA. So we should be aware about these types of nucleotides, types of nucleosides, and the examples. I think with this, it is very easy to remember using these formulas. So nucleotides are also called as base sequences. Okay, so the nucleotides are heterocyclic compounds. So nucleic acids, DNA and RNA are made up of nucleotides. So nucleic acids are polynucleotides, we call it as. So nucleic acids are polynucleotides. So DNA and RNA are made up of nucleotides. So you can notice this nitrogenous bases, how they form this DNA. So in DNA, the sugar would be deoxyribose. So deoxy, one oxygen would be absent, whereas in RNA it would be ribose sugar. So you can notice here the adenine is given different coloring, guanine is given different coloring, thymine is green in color, cytosine is light blue in color, guanine is pink in color, adenine is orange in color. Okay, so the adenine always base pairs with thymine, or we can, in case of RNA, adenine base pairs with uracil. In case of DNA, adenine always base pairs with thymine. So you can notice that wherever the orange color is there, you notice that they would be having the uh, green colored attachment of it. The red one, so you find the green colored attachment. That is adenine always base pairs with thymine. Okay, and the guanine always base pairs with cytosine. So the base pair of adenine with thymine is a double bond. The base pairing of adenine with cytosine, sorry, the guanine with cytosine is triple bond. Okay, and vice versa. So you should remember about this. So they have a nitrogenous base sugar, you can notice here. So the nucleotide consists of the nitrogenous base sugar and phosphate group. So you notice the sugar here which has this nitrogenous base and phosphate group. So the sugar here is deoxyribose, so DNA, and you can see in between these So there are phosphodiester bonds between the nucleotides. So the phosphodiester's bonds are noticed between the nucleotides. Okay, so nucleotides are heterocyclic compounds. They are heterocyclic compounds. You can notice here. So nucleotides are heterocyclic compound. Nucleic acids, that is DNA and RNA, they are made up of many nucleotides or polynucleotides, you can call it as. So nucleic acids, DNA and RNA are made up of many nucleotides, or we also call it as polynucleotides. So after completion of biomicromolecules, we'll go to the next topic, that is biomacromolecules. Okay. See, biomacromolecules are molecules having molecular weight greater than 1000 Daltons. They include proteins, polysaccharides, nucleic acids. So biomicromolecules we had discussed about amino acids, sugars, and nucleotides. Okay, so nitrogenous bases we had discussed about, but in biomacromolecules we are going to discuss about proteins, polysaccharides, and nucleic acids. So their molecular weight is 10,000 Daltons and above. So I told it as greater than 1000 Daltons, but generally the biomacromolecules, their molecular weight is 10,000 Daltons and above. So they are acid insoluble fraction. So macromolecular fraction. So they are acid insoluble fraction, which includes macromolecules from cytoplasm and organelles. So they are acid insoluble fraction and they will be as the remnants. Okay, so they are not the filtrate, so they would be found as retentates. These biomolecules, macromolecules. So because they are acid insoluble fraction, so which we also call it as macromolecular fraction. So they include macromolecules from cytoplasm and cell organelles. So the macromolecules, they include proteins, polysaccharides, and nucleic acids. So we'll be studying about this proteins, polysaccharides, and nucleic acids in the present class. Already you know the structure of the cell membrane, the fluid mosaic model of plasma membrane. So you notice the proteins which are the superficial proteins, as well as this proteins which are integrated in the membranes. So the peripheral proteins and the integrated proteins, you notice that, and most of the proteins are floating within this phospholipid bilayer. Okay, this is what you notice in case of the fluid mosaic model of plasma membrane or cell membrane. So lipid is not strictly a macromolecule, as its molecular weight does not exceed 800 Daltons. And we have already studied lipid and types of lipids in case of biomicromolecules. But the lipids comes under acid insoluble fraction because many lipids are arranged into structures like cell membranes. When a tissue is grinded, cell membranes are broken and they form water insoluble vesicles. They cannot be filtered along acid soluble fraction. Even though lipid is not strictly a macromolecule, as their molecular weight does not exceed 800 Dalton. So it comes under the acid insoluble friction because many lipids are arranged into structures like cell membrane. So the lipids are arranged into structures like cell membranes. When a tissue is grinded, the cell membranes are broken and they form water insoluble vesicles, which cannot be filtered along the acid soluble fraction. So the average composition of cells, if you look into these composition of cells and cytoplasm, water is 70 to 90%, protein is 10 to 50%. So the component of a cell protein is 10 to 15%. Carbohydrates, it's very less, 3% of it. Nucleic acid is 5 to 7%, and the ions are for 1%. So the average composition of cells or cytoplasm, if you look into, the water is 70 to 90% of the cell is composed of water. Proteins are 10 to 15%. The carbohydrates are 3%, and nucleic acids are 5 to 7%. Ions are 1%. So this is what you have to remember about the average composition of a cell or its cytoplasm. Okay. So now we'll discuss about the first biomicro molecule, biomacromolecule rather, proteins we'll be discussing about. Okay, so the proteins are heteropolymer of amino acids. So they are having a heteropolymer. It's not a mono homopolymer, it is heteropolymer. That implies they might have different types of amino acids, not the same type of amino acids. So that is the reason we use the term proteins are heteropolymer of amino acids. If they are made up of same type of amino acids, so we call it as homopolymer of amino acid, or you can just say it as polymer of amino acids. But proteins are made up of different types of amino acids. That is the reason we use the term heteropolymer of amino acids. So you can notice here different types of amino acids, they are going to form the proteins. So A1, A2, A3, A4, A5, A6. So different types of amino acids. So it's a heteropolymer of amino acids. Okay, and they are polypeptides, that is linear chains of amino acids linked by peptide bonds. So the proteins are also called as polypeptides since they are linear chain of amino acids which are linked by peptide bonds. So we call it as polypeptides. Okay, so they are linear chain of amino acids which are linked by peptide bonds. So that is the reason we call this proteins also has polypeptides. See here in the structure of the protein, you can notice that pink colored is one type of amino acid and blue colored is another type of amino acid. So they are heteropolymer of uh, they are heteropolymer of amino acids. So proteins are also called as polypeptides, that is linear chains of amino acids which are linked by peptide bonds. So peptide bond is formed when the COOH group of amino acid reacts with, see peptide bond is formed when COOH group of amino acid uh, reacts of COOH group of one amino acid reacts with NH2 group of next amino acid by releasing a molecule of water, that is dehydration takes place. So that is how the peptide bond is formed. See here you can notice that peptide bond formation. So peptide bond is formed when COOH of one amino acid eh, reacts with amino group of next amino acid by releasing a molecule of water. So they form a bond here. So there is a dehydration here. So H2O is released. One NH, one H from NH2 and OH from COOH. They combine to form water and there is dehydration. So a molecular of water is released, and this carboxylic group of one amino acid which gets attached to the amino group of another amino acid to form a peptide bond. You can notice here by the release of water or dehydration. So these peptide bonds are formed. So the proteins are also called as polypeptides, that is linear chain of amino acids. So linked by peptide bonds. Okay. So peptide bond is formed when COOH of one amino acid, it reacts with NH2 group of amino acid by releasing a molecule of water. See this H2O is lost and there is a bond formed between these two amino acids which we call it as peptide bonds. That is how the polypeptides are formed. So now the functions of proteins. So for growth and tissue repair, proteins are required. So the growth and tissue repair, we require the proteins. So then there is transport of nutrients across cell membrane. So example, so you know that some of the proteins they act as carrier proteins. So the transport of nutrients across cell membranes is also brought by proteins. Proteins bring about transport of nutrients across cell membranes. Example is GLUT4 enables glucose transport into cell. So proteins are used for growth and tissue repair. So you know that nearly a cell 10 to 15% of the composition of a cytoplasm is protein. So they are very important for growth as well as tissue repair. So proteins transport nutrients across the cell membranes as carrier proteins. You know that example is GLUT4 enables glucose transport into cell. So they transport nutrients across cell membranes. So proteins also acts as intercellular ground substance. So proteins also acts as intercellular ground substance. Example is collagen. So proteins acts as antibodies to fight infectious organisms. See, antibodies are proteinaceous in nature. So they act as antibodies to fight infectious organisms. Okay, so antibodies, or we also call it as immunoglobulins. So the antibodies are proteinaceous in nature, enzymes are proteinaceous in nature, hormones are proteinaceous in nature. So the proteins they act as antibodies to fight against infectious organisms. So the proteins also acts as a receptor. So example is the receptor of smell, taste, and hormones are by the proteins. Proteins also acts as receptors. Example is receptors of smell, taste, and receptors of hormones. So proteins also acts as receptors. The various receptors like receptors of smell, receptors of taste, receptors of hormones. Some of the hormones, example insulin, enzymes, example trypsin, pigments like hemoglobin, they are all proteinaceous in nature. Some of the hormones like insulin, some of the enzymes like trypsin, some of the pigments like hemoglobin, they're all proteinaceous in nature. So you should remember about this proteins, their various functions of the proteins. So proteins are required for growth and growth and tissue repair. So proteins transport nutrients across cell membranes. Example is GLUT4, which enables glucose transport into the cell. So proteins acts as intercellular ground substance. Example is collagen. Collagen is also proteinaceous in nature. So the proteins also act as antibodies. So antibodies are proteinaceous in nature. Proteins also acts as antibodies to fight against infectious organisms. So proteins acts as various receptors. Example, they act as receptor of smell, receptor of taste, and receptor of hormones. Some of the hormones like insulin, enzymes like trypsin, and pigments like hemoglobin, they are all proteinaceous in nature. So you should remember about this insulin, trypsin, hemoglobin, they are proteinaceous in nature. So some of the hormones, enzymes, and hemoglobin pigments, they are all proteinaceous in nature. So proteins, most abundant protein in animal world is collagen. Most abundant protein in the entire biosphere, if you take, is rubisco. Ribulose biphosphate carboxylase oxygenase. So remember about this Rubisco enzyme, which we'll be studying in the photosynthesis. Okay, they help in fixing carbon dioxide. The Rubisco enzyme helps in fixing the carbon dioxide. Rubisco enzyme plays an important role in C3 cycle. So when we come to plant physiology, photosynthesis, we'll be discussing about this rubiscus. In proteins, the most abundant protein in the animal world is collagen. In the animal world, the most abundant protein is collagen. Whereas the most abundant protein, if you take the entire biosphere, the most abundant protein in the biosphere is the rubisco enzyme, that is ribulose biphosphate carboxylase oxygenase enzyme. Rubisco, you can remember it as that Rubisco. Okay, the enzyme which is involved in photosynthesis, or that is especially the C3 cycle or Kelvin cycle. So the enzyme which fixes the atmospheric carbon dioxide is Rubisco enzyme. So that is the most abundantly found protein in the biosphere is Rubisco enzyme. So ribulose biphosphate carboxylase oxygenase enzyme. Okay. So on the structural level of proteins, proteins are of different types, different structural level, the primary structure, secondary structure, tertiary structure, and quaternary structure. Structural level of proteins, the proteins are having primary structure, they are having secondary primary structure or secondary structure or tertiary structure or quaternary structure. Based on the structural level of proteins, proteins might have either primary structure or secondary structure, or they might be having tertiary structure or quaternary structure. So primary structure, when we discuss about, so it describes the sequence of amino acids, that is the positional information in a protein. So what are the types of protein and what are their positions? That is described by the primary structure. It describes the position of the sequence of amino acids, that is positional information in a protein. It describes the sequence of amino acids, which type of amino acid sequence they have. It describes that. So primary structure always describes the sequence of amino acid. That is, it provides positional information in a protein. Okay. Left end, that is first amino acid, N terminal amino acid it is called as. Right end, last amino acid is called as the C terminal amino acid. So the first end amino acid, first amino acid, left end is the first amino acid, that is N terminal amino acid. Right end is the last amino acid with C terminal amino acid. That is, they have the carboxylic group. N terminal amino has the amino group there. So the left end is the first amino acid, that is N terminal amino acid. Right end is the uh, last amino acid, that is the C terminal amino acid, which has this COOH group here. So remember about that, left end and right end, on what basis we are going to consider them is the first left hand will be having the N terminal amino acid. That is the first amino acid which will be having this N terminal amino acid. So the right hand, it will be having the last amino acid, that is C terminal amino acid. So the primary structure, it describes the sequence of amino acids. That is, it provides positional information of protein. Like here you notice the R1, R2, R3, R4. So the positional information of a protein is given by the primary structure. So in the secondary structure, see, these simple diagrams are required, but don't worry about these diagrams which are, they're not going to ask you these diagrams in your examination. So please don't worry about it. Neither they ask you the structural formulas, but when you look at of a structure of a formula of a amino acid or a nucleic acid, you should be able to identify them, whether they are nitrogenous base, whether they are nucleoside or nucleotide. You should be able to identify them and label them. So that is the thing that you should be developing those skills, but you need not have to mug up the structures unless and until I've told you about it. Okay, so maybe some of the molecular formulas might be required, but structural formula is not required in the biology part of this biomolecules. So try to understand the concepts with the diagrams that we have here. So the secondary structure, here one or more polypeptide chains are folded in the form of a helix. So here one or more polypeptide chains, they are folded in the form of a helix. So you can notice here, so the beta pleated sheet, then helix, alpha helix. So one or more polypeptide chains, They are folded in the form of a helix. It has only right-handed helices. So there are one or more polypeptide chains, they are folded into a helix. It has only the right-handed helices. Example, keratin, fibroin, which is noticed in silk fiber. They exhibit this secondary structure with right-handed helices. So they have the right-handed helical structures. So we call it as right-handed helices. So here one or more polypeptide chains are folded in the form of a helix. So it has only right-handed helices. Example is keratin, fibroin. Fibroin is nothing but the silk fiber. So typical strand of hair, keratin, which is found in the hair. So they have this helical structures and they are right-handed helices that you can notice in keratin. So they have a strong disulfide bonds. Weak links are noticed there. So they have this one or more polypeptide chains which are having this helical structures. Okay, they are folded in the form of a helix. So keratin is an example of a typical strand of hair having keratin. You can notice the secondary structure. Then fibroin, the silk protein, you can notice this anti-parallel beta sheet rich in uh, alea and glycine, alanine and glycine, small chains. So you can notice this alanine and glycine, small chains, which are small side chains. They allow close packing. So alanine and glycine. So you notice the small chains which allow the close packing. Okay. This is noticed in case of fibroin, the silk protein. So they also exhibit this secondary structure. Not required for your examination, but just to make you understand the helical structure of this one or more polynucleotide chains, polypeptide chains. They are folded in the form of a polypeptide chains are folded in the form of a helix. So in case of example is keratin as well as fibroin in silk protein, you notice this helical structure of the polypeptides. Then the tertiary structure we'll discuss about. Here helical polypeptide chain is further folded like a hollow woolen ball. So they are going to be further folded to form a hollow woolen ball. So it gives a 3D view, three-dimensional view you can see in case of tertiary structure. So tertiary structure is necessary for many biological activities of proteins like myoglobin, enzymes, they have this tertiary structures. So the helical polypeptide chain is further folded to form a hollow means there is a space, hollow woolen ball. So the helical polypeptide chain is further folded to form a hollow woolen ball. So it gives a 3D view. So they are further folded, the polypeptide chains are further folded, helical polypeptide chains to form a hollow woolen ball. It gives a 3D view or three-dimensional view. Tertiary structure is necessary for many biological activities of proteins like myoglobin, enzymes. They're all examples for tertiary structure. They are required for many biological activities of proteins. So the myoglobin and enzymes are an example for tertiary structure. Then the quaternary structure, what you notice here is more than one polypeptide chain forms tertiary structure, and each chain functions as subunits of protein. So more than one, see you can notice here in this quaternary structure, more than one. There is one quaternary structure which is blue in color. There is another quaternary structure which is green in color. And there is one more quaternary structure which is pink in color. So more than one quaternary structure, one more than one polypeptide chain form the tertiary structure, and each chain functions as subunits of proteins. Here in quaternary structure, you notice that more than one polypeptide chains they form tertiary structure, and each chain functions as subunit of proteins. Example, hemoglobin, it has four subunits. So two alpha subunits, you can notice two alpha subunits. so the bluish one, and two beta subunits. So here you find more than one polypeptide chain, they form tertiary structure. So more than one polypeptide chain, they form the tertiary structure, and each chain function as subunits of protein. Like in case of hemoglobin, you notice they have four subunits, two alpha uh, subunits and two beta subunits. So more than one polypeptide chain, they form tertiary structure, and each chain, they are going to function as subunits of protein. So example, hemoglobin, it has four subunits, of which two are alpha subunits and two of them are beta subunits. So each one of them forms a tertiary structure, and there is more than one tertiary structure in case of hemoglobin, and they form subunits of protein. So this is the quaternary structure. So more than one subunit, more than one polypeptide chain, they form the tertiary structure, and each chain function as subunit of proteins. That is nothing but the quaternary structure of proteins. Example is hemoglobin. It has four subunits, two alpha subunits and two beta subunits are noticed. So the structural level of proteins, at a glance, you can notice that they have a primary structure of amino acids. It's a polynucleotide, uh, polypeptide, not polynucleotide, it is polypeptide chains. The secondary structure, you find that they are arranged in an helical structure, more than one polypeptides, they are arranged in a helical structure arrangement. In tertiary, the helical structures are further folded to form hollow ball-like structure. Okay, so which looks like a hollow woolen ball-like structure. So with a hollow central cavity of woolen ball-like structure, you notice that. So more, and they are further folded to form a hollow woolen ball-like structure, and you notice that they are three-dimensional structures, which you can notice. If they are having more than one, uh, more than one polypeptide chain which exhibits the tertiary structure, so we call it as the quaternary structure. So such polypeptides which are exhibit more than one tertiary structures are exhibited in a uh, unit. We call it as the quaternary structure. Example is hemoglobin, where they have four subunits, two alpha subunits and two beta subunits. So primary structure will have a chain of amino acids. Secondary structure, they have alpha helical like structures, okay, or pleated sheet they might have, as in case of fibroin. Then alpha helical structure, you notice it. Okay, so then the tertiary structure, which has this, the further, the helical structure is further folded to form a woolen ball like, hollow woolen ball like unit. So the three-dimensional unit, we call this as tertiary structure. So some of the enzymes, they exhibit this sort of tertiary structures. So quaternary structure, they have more than one uh, tertiary structures are seen in this polypeptide chain, and they form the subunits of proteins. We call them as quaternary structure. Hemoglobin is an example where they have two alpha and subunits and two beta subunits. Okay, so the next macromolecule we'll discuss is polysaccharide. So polysaccharide, these are polymers of sugars, or you can also call it as polymers of monosaccharides. Example, starch. It is homopolymer of glucose. Starch is a homopolymer of glucose. Cellulose is a homopolymer of glucose. Okay, glycogen is also homopolymer of glucose, the reserve food material in animals, especially they are stored in the liver and muscles. Starch is a reserve food material in plants, but they are homopolymer of glucose. They are stored in plant starch. So cellulose is also homopolymer of glucose. Cellulose, you find it in the cell wall. The glycogen is also reserve food material stored in animals, which is a homopolymer of glucose. Inulin, which is found in the flowers and others. So the inulin is homopolymer of fructose. Okay, fructose, you know that it's an isomer of glucose. So they have the same chemical formula, but structurally they are different. So isomers are having the same chemical formula, but structural formula they vary. Okay. So the polysaccharides, these are complex polysaccharides formed of amino acid. Example, polysaccharides, these are complex polysaccharides formed of amino sugars. Example is glucosamine, N-acetyl galactosamine. So they're all complex polysaccharides. Chitin is the homopolymer of N-acetyl glucosamine. It is seen in the exoskeleton of arthropods and fungal cell wall. So chitin is what? It is the homopolymer of N-acetyl glucosamine. It is seen in the uh, exoskeleton of fungal cell wall as well as the arthropods, exoskeleton of arthropods and the fungal cell wall. They are made up of chitin. What is chitin? It is a homopolymer of N-acetyl glucose amine. Okay, so you can notice that in case of this fungal cell wall, N-acetyl glucosamine. So the chitin is made up of this N-acetyl polymer of N-acetyl glucosamine. In the arthropods, the exoskeleton is made up of N-acetyl homopolymer of N-acetyl glucosamine. When we say homopolymer, the monomers are of the same type. That is why we use the term homopolymer. Okay, so polysaccharides, there are complex polysaccharides which are formed of amino sugars like glucosamine, N-acetyl galactosamine. So amino sugars we call them as with amino acid and sugars. So chitin is the homopolymer of N-acetyl glucosamine. It is the seen in the exoskeleton of arthropods and the fungal cell wall. The polysaccharides, the glycosidic bonds in polysaccharides, you can notice. So two monosaccharide subunits are there. So they have this OH group. So two monosaccharide subunits, monomers. So they combine to form the glycosidic linkage. So OH, they lose from the OH of both these monomers, they lose a molecule of water and they form a glycosidic linkage. So the oxygen is linking them. So the glycosidic linkage you can notice in case of the uh, polysaccharides. So glycosidic bond holds the monosaccharides together to form the polysaccharides. So two monosaccharide subunits they fuse to form with the loss of water they form this glycosidic linkage which is held by the oxygen. So glycosidic linkage, it is the bond formed when individual monosaccharides are linked between two carbon atoms. So individual monosaccharides, if they are linked between two carbon atoms by dehydration, so we call that as a glycosidic linkage. So this is the diagrammatic representation of a portion of glycogen. So how they look like branched and rebranched. So that is the portion of this glycogen is shown. So enlarged and shown for you to understand it better. On this main chain, it would be of this sort of arrangement of glycoside. So there are side chains also in the case of the glycogen. So they branch and again branch. So this sort of pattern of arrangement you notice in case of glycogen. So diagrammatic representation of a portion of glycogen. So they are all held by this glycosidic bonds between the two carbon atoms of monosaccharides. They lose water. Okay. So that sort of bond formed is glycosidic bonds. You can notice that everywhere there is a glycosidic bonds that are formed. So starch is going to form helical secondary structures. So you can notice in case of starch, they are having this helical secondary structure. It can hold L2 molecules in the helical portion, giving the blue color. So it can hold this L2 molecules in the helical portion, giving the blue color. Okay, so cellulose has no complex allysis and therefore cannot hold L2. So they do not have this complex helical structure. So therefore, they cannot hold this L2. So that is how you differentiate between starch and cellulose. So the starch, when they are treated with iodine, they form the blue color. So that is because of this L2 molecules in the helical portions are seen, whereas the L2 molecules in cellulose are not seen because they do not have this complex lysis. So the L2 molecules in the helical portion is responsible for the blue color whenever they are tested with iodine, whereas cellulose has no complex helical helices and therefore they cannot hold this L2 molecules. So those L2 molecules are not there. As a result, they don't react with iodine to produce blue color. Okay, so I think we will stop with the polysaccharides. In the next class, the biomacromolecules of nucleic acid we will discuss about. Okay, so you have any doubts or clarifications on this, dear students?